Basic knowledge about vaccines is very important in AEFI surveillance, because in-depth investigations of reported AEFIs include inquiries of the vaccine involved. Therefore, health workers involved in AEFI surveillance should have basic knowledge of vaccines. After watching this video, you should be able to define vaccine, describe an ideal vaccine, and enumerate the types of vaccines. A vaccine is a preparation that is used to stimulate the body's immune response against diseases. So, the primary requirement for a preparation to be considered as vaccine is that it should stimulate the body's immune response. Therefore, by this definition, do you consider human rabies immunoglobulin a vaccine? Since human rabies immunoglobulin does not stimulate the body's immune response against rabies, it is not considered a vaccine. So, it is simply a medicinal, biological preparation.
Doc Marl, ano ang mga pamantayan na dapat kong gamitin upang matukoy kung ang isang bakuna ay may magandang kalidad?
Several vaccines have been introduced against various diseases across the world. But in this ocean of vaccines, one must thoroughly be acquainted with the properties of an ideal vaccine before being subjected to it. The two most important desirable features in a vaccine to qualify it as an ideal vaccine are safety and efficacy. The most important feature that demands for substantial scrutiny in any vaccine is its safety. Initial testing of any vaccine involves testing in animals. Animal testing is critical to ensure that the vaccine does not end up making the infection worse or even fatal. Preclinical animal toxicity studies are conducted before the start of human trials. Testing in animals also helps researchers understand the type of immune response that will be generated in a vaccine and whether that might be enough to protect the individual from developing the disease, known as the efficacy. If laboratory and animal studies indicate the vaccine is safe, additional testing in people is done before the vaccine is licensed for public use. We call this clinical trial. The trial has 4 phases. Phase 1 of the trial aims to determine serious side effects, dosing, and dose-related side effects. It involves only few volunteers. The second phase continues to explore possible side effects of the vaccine and how the immune system is responding to the vaccine, meaning its efficacy. In phase 3, researchers explore how well can the vaccine protect people from the disease. This is done by comparing the disease rates between people who get the vaccine and those who do not. If phase 3 is successful, the sponsor applies for new drug application to the Food and Drug Administration. The FDA will review the safety and efficacy of the vaccine and assess whether the vaccine's benefits outweigh the risks. If the review is favorable, the FDA will approve the vaccine for marketing or public use. After FDA approval, phase 4 of the trial begins. Phase 4 is conducted after the vaccine is already marketed and available to the general public. The main objective is to check the vaccine's performance in real-life scenarios and discover any rare side effects. This can last for many years. So, AEFI surveillance is part of phase 4 of the clinical trial. This is the reason why we give so much importance to this activity.
Doc Marl, maaari mo bang ipaliwanag sa akin ng higit pa kung ano ang ibig sabihin ng benefits outweigh the risks?
Good you ask that question, Rosie. Just like any medicine, when a vaccine is considered safe, it does not mean it is entirely risk-free. So, when evaluating a vaccine, it is important to consider both the benefits and risks, including both the risk of receiving the vaccine and of not receiving them. The benefits of a vaccine are its positive effects, like reducing death, reducing hospitalization, or protecting people from serious illness. The risks are the unwanted or unexpected effects that can span from relatively minor to rare, or more serious events. Therefore, a quality or safe vaccine should have benefits that outweigh or exceed the risks, otherwise, the vaccine will not be approved for public use. The benefits and risks of a vaccine are explored or discovered in a clinical trial we have discussed earlier.
Salamat Doc Marl. Ngayon ko lang lubos na na-appreciate ang kahalagahan na maintindihan ang pagsusuri sa benepisyo kumpara sa panganib ng mga bakuna. Doc Marl, ano naman yung vaccine efficacy? Ano ang kaibahan nito sa vaccine effectiveness?
Vaccine efficacy is the second most important desirable feature of an ideal vaccine. Vaccine efficacy refers to how the vaccine performs during a clinical trial. It is based on how many people who got vaccinated developed the disease, compared with how many people who got the placebo or dummy vaccine developed the same outcome. So, for example, in a clinical trial, 2 of the 10 volunteers who received the vaccine got ill, compared to 9 of the 10 volunteers who received the placebo got ill. Based on this result, the efficacy of the vaccine is 8 out of 10, or 80%. This means that those who received the vaccine were prevented of developing the disease by 80% compared to the group who received the placebo, which was only 10%. Just to reiterate, efficacy is the degree to which a vaccine prevents disease under ideal and controlled circumstances in a clinical trial, comparing a vaccinated group with a placebo group. To be approved, vaccines are required to have a high efficacy rate of 50% or above. If vaccine efficacy refers to how the vaccine performs during a clinical trial, vaccine effectiveness refers to how well a vaccine performs in the real world. A vaccine may have a high efficacy rate during a clinical trial, but low effectiveness when applied to the general population because of different influencing factors. Now, I have a question for you. Immunization surveillance data is vital to understanding effectiveness of vaccines. Is the statement true or false? The answer is true. In addition to observational studies, immunization surveillance data is vital to understanding effectiveness of vaccines. Remember, vaccines do not always need to have an exceptionally high effectiveness to be useful. For example, the influenza vaccine is only 40 to 60% effective, yet it saves thousands of lives every year. Now, let's move on to our next subtopic, the types of vaccines. Vaccines may be monovalent, polyvalent, or combined. A monovalent vaccine contains a single strain of a single antigen, or microorganism. An example to this is measles vaccine. Whereas, a polyvalent vaccine contains 2 or more strains, or serotypes, of the same microorganism. An example to this is pneumococcal vaccine. Pneumococcal polyvalent vaccine contains highly purified capsular antigens from the 23 most prevalent or invasive pneumococcal types of Streptococcus pneumoniae to ensure cross-protection. Combined vaccines contain 2 or more antigens. An example to this is the MMR vaccine. The potential advantages of combined vaccines include reducing the cost of stocking and administering separate vaccines, reducing the cost of extra healthcare visits, improving timeliness of vaccination, and facilitating the addition of new vaccines into immunization programs. Vaccines are usually administered through needle injections, but some can be administered by mouth or sprayed into the nose.
Doc Marl, totoo ba na ang pagbibigay sa isang bata ng combination vaccine gaya ng MMR ay nagpapataas ng panganib ng mga mapaminsalang epekto at maaari itong mag-overload sa immune system ng bata?
The moment babies enter the world, they encounter millions of foreign organisms. The infant's immune system is no longer considered immature, but is finely tuned to respond to the incredible number of viruses, bacteria, and other organisms it meets early in life. Vaccines contain just a few antigens, or organisms, compared to what babies meet every day. Infant immune systems are stronger than you might think. Even if all 14 scheduled vaccines were given at once, it would only use up slightly more than 0.1% of a baby's immune capacity. Therefore, combined vaccines are as effective as the single injection, and they do not pose any safety concerns or greater risk to the child. The main types of vaccines that act in different ways are the following: live attenuated vaccines, inactivated vaccines, subunit vaccines, toxoid vaccines, mRNA vaccines, and viral vector vaccines. The first type of vaccine is live attenuated vaccine. Live attenuated vaccines contain whole bacteria or viruses which have been weakened or attenuated so that they create a protective immune response, but do not cause disease in healthy people. This weakening is achieved through genetic modification of the pathogen, either as a naturally occurring phenomenon or as a modification specifically introduced by scientists. The immune response to a live attenuated vaccine is virtually identical to that produced by a natural infection, because the immune system does not differentiate between an infection with a weakened vaccine virus and an infection with a wild virus. Because of this, live attenuated vaccines create strong and long-lasting immune response. Just 1 or 2 doses, these vaccines can give lifetime protection against the disease it causes. However, live attenuated vaccines may not be suitable for people whose immune system doesn't work, either due to drug treatment or underlying illness. This is because the weakened viruses or bacteria could in some cases multiply too much and might cause disease in these people. On the other hand, the weakened virus may change over time and behaves more like the wild or naturally occurring virus. This means it can be spread more easily and may cause illness. An example to this is OPV or oral polio vaccine. The live attenuated polioviruses may change over time and behaves like a wild poliovirus that can cause VAP or vaccine-associated paralytic poliomyelitis. For this reason, the global eradication of polio requires stopping all OPV and routine immunization as soon as possible after the eradication of wild poliovirus transmission in a country. In these countries, OPV will be replaced with an injectable, inactivated polio vaccine or IPV. The other limitation of live attenuated vaccines is they need to be stored at a temperature of 2 to 8 degrees centigrade. That means they can't be used in countries with limited access to refrigerators. Examples of live attenuated vaccines are the following: OPV, MMR, BCG, and rotavirus. The second type of vaccine is inactivated vaccine. An inactivated vaccine is a vaccine consisting of virus particles, bacteria, or other pathogens that have been grown in culture and then killed to destroy their disease-producing capacity. The inactivation step usually involves heat, radiation, or chemicals to destroy the pathogen's genetic material, which stops it from replicating. Inactivated vaccines require special laboratory facilities to grow the virus or bacterium safely, and can have a relatively long production time.
Alam kong inactivated vaccine ang Sinovac. Doc Marl, bakit kailangan pa ng booster doses ang mga taong nabakunahan ng Sinovac? By the way, ang tamang pronunciation ng word na ay Sinovac.
Unlike live attenuated vaccines, which produce an immune response that closely resembles natural infection, the immune response to an inactivated vaccine is mostly antibody production. Little or no cellular immunity results. Antibody titers against inactivated antigens diminish with time. As a result, some inactivated vaccines may require periodic supplemental doses to increase or boost Antibody titers. Because inactivated pathogens tend to produce a weaker response by the immune system than live attenuated vaccines, adjuvants are added in some vaccines to enhance its capability of activating the body's immune response against the pathogen. Some people who cannot take live attenuated vaccines, like the elderly and those with immunodeficiency, an inactivated vaccine is suitable for them. Examples of inactivated vaccines are the following: inactivated poliovirus vaccine, hepatitis A vaccine, rabies vaccine, and Sinovac. The third type of vaccine is the subunit vaccine. A subunit vaccine is a vaccine that contains purified parts of the pathogen that are antigenic or necessary to elicit a protective immune response. A subunit vaccine doesn't contain the whole pathogen, unlike live attenuated or inactivated vaccine, but contains only the antigenic parts of the pathogen, such as proteins, polysaccharides, or peptides. The advantages of subunit vaccines are the following. First, subunit vaccines are very safe because they don't contain live organisms. Second, it is a well-established technology. Third, they are suitable for immunocompromised individuals. The disadvantages are the following. First, they are relatively complex to manufacture compared to some vaccines. Second, they require adjuvants and booster shots. Third, they require time to examine which antigenic combinations may work best. Lastly, the immune response may only be antibody-mediated and not cell-mediated, because the antigens of subunit vaccines do not infect cells. Therefore, subunit vaccines are weaker than those elicited by live attenuated vaccines. Examples of subunit vaccines are the following: hepatitis B vaccine, pneumococcal polysaccharide vaccine, and meningococcal vaccine.
Doc Marl, ano naman yung conjugate vaccine? Magkaiba ba ito sa subunit vaccine?
Let me describe a conjugate vaccine before I will give its definition. Bacteria are coated by a sugar called polysaccharide. A portion of this coating is removed and injected into humans as a form of vaccine. However, scientists have observed that this pure polysaccharide vaccine did not generate a strong immunity against the bacteria, meaning it's a weak antigen. However, when the polysaccharide was attached or connected to a tetanus toxoid, a strong antigen, it generated a strong immune response against the pathogenic bacteria. Therefore, a conjugate vaccine is a type of subunit vaccine which combines a weak antigen with a strong antigen as a carrier, so that the immune system has a stronger response to the weak antigen. The advantages of conjugate vaccines are the following. First, the vaccine elicits strong antibody and cell-mediated immune response among infants and children. Second, it helps reduce mucosal carriage of pathogenic organisms, resulting to enhanced herd immunity. Lastly, it offers long-term protection. However, conjugate vaccines are expensive. Other disadvantages are the same as for subunit vaccines. Examples of conjugate vaccine include Haemophilus influenza conjugate vaccine and pneumococcal conjugate vaccine.
Doc Marl, ano naman yung recombinant vaccine? Magkaiba ba ito sa conjugate vaccine?
I will simplify the definition of a recombinant vaccine by describing to you how hepatitis B vaccines are made using recombinant DNA technology. First, a small piece of DNA or genetic material is taken from hepatitis B virus. Then, a small circular DNA molecule, called plasmid, is taken from a bacterium. Then, the small piece of hepatitis DNA is inserted into the plasmid DNA of the bacterium. The combination of these two DNAs is now called a recombinant DNA. Then, the recombinant DNA is injected into yeast cells. These yeast cells become recombinant yeast cells. When recombinant yeast cells are placed in a fermentation tank, they multiply and produce hepatitis B surface antigens. These antigens are purified and used as the active ingredient of hepatitis B vaccines. Therefore, a recombinant vaccine is a type of subunit vaccine that is made of joined DNA molecules and inserted into host organisms that produce antigens of the pathogenic organism. Recombinant vaccines are safer than attenuated or inactivated vaccines. They can be easily produced compared to the traditional vaccines. They are less expensive. Therefore, recombinant vaccines may offer a better solution in the long term. Just like other subunit vaccines, they need for repeated vaccination and use of adjuvants to boost their immunogenicity. Examples of recombinant vaccines are hepatitis B vaccine and human papillomavirus vaccine. The fourth type of vaccine is toxoid vaccine. Some bacteria release toxins or poisonous proteins when they attack the body. It's the toxin that causes illness rather than the bacteria itself. For example, the bacteria Clostridium tetani, when inside our body, produce poisonous toxin called tetanospasmin. It's the toxin tetanospasmin that causes the disease tetanus, rather than the bacteria itself. Scientists develop toxoid vaccines that can neutralize these toxins in our body. Toxoid vaccines are made of inactivated toxins, derived from toxins that are secreted by certain species of bacteria. They are called toxoids because they look like toxins, but are not poisonous. They trigger a strong immune response once injected into the body. Examples of toxoid vaccines are tetanus toxoid and diphtheria toxoid. The fifth type of vaccine is the mRNA vaccine. An mRNA vaccine is a new type of vaccine which contains the genetic material of a specific part of the virus encoded in a synthetic protein called mRNA. The synthetic mRNA is packaged within a lipid vector called nanoparticle. mRNA vaccines do not contain dead or weakened virus. It does not contain even a small part of the virus. It has only millions of artificial or synthetic mRNA proteins that contain the formula of a specific protein of the virus. Now, when the vaccine is injected into your upper arm muscle, the mRNA nanoparticles enter into your muscle cells. Then, the muscle cells read the formula or code from the synthetic mRNA and produce millions of copies of viral proteins that are displayed on the surface of the muscle cells. The displayed viral proteins are recognized by the immune system, which produces specific antibodies and T cells to attack them. The advantages of mRNA vaccines are the following. First, mRNA vaccines do not contain live virus and carry no risk of causing disease in the vaccinated person. Second, faster manufacturing time, because synthetic mRNA molecules are easier to produce in the laboratory. Third, mRNA vaccines can activate both the antibody and cell-mediated immunity. Lastly, there is no risk of mRNA integrating into host DNA. The disadvantages are the following. First, mRNA vaccines must be kept at very low temperatures, between -80 and -60 degrees Celsius, to avoid degrading, and thus giving little effective immunity to the recipient. Second, may cause strong immunogenicity, triggering unnecessary immune response. Lastly, its long-term side effects and duration of protection are still unknown. Examples of mRNA vaccines are Pfizer and Moderna vaccines. The sixth type of vaccine is viral vector vaccine. Let me describe to you how a COVID-19 viral vector vaccine is made. First, a genetic material is extracted from the spike protein of the COVID-19 virus. Second, the extracted spike protein gene is inserted into the gene of a harmless adenovirus. The virus is harmless because it cannot replicate, therefore, loses its ability to cause disease. The adenovirus now becomes a viral vector, meaning it serves as a vehicle to deliver the COVID-19 spike protein gene to the cells of our body. Once the viral vector vaccine is injected into the upper arm muscle, the adenovirus that contains the COVID-19 spike protein genetic code invade the muscle cells and releases the genetic code. Then, The muscle cells read the genetic code and produce millions of copies of spike proteins displayed on the surface of the muscle cells. The displayed spike proteins are recognized by the immune system, which produces specific antibodies and T cells to attack them. Therefore, a viral vector vaccine is a vaccine that uses a harmless virus as vehicle to deliver genetic material coding for a desired antigen into the recipient's host cells. Viral vector vaccines can trigger strong antibody and cell-mediated immune system responses that are anticipated to provide longer-term protection. Viral vector vaccines offer operational benefits, including a single-dose product series and less stringent storage and handling conditions. A major bottleneck for viral vector vaccine production is scalability. Assembling the vector vaccine is also a complex process involving multiple steps and components, each of which increases the risk of contamination. The other limitation of viral vector vaccines includes diminished immune responses and vaccine effectiveness in individuals with pre-existing immunity to the viral vector. However, this limitation is overcome for COVID-19 vaccines by using adenovirus serotypes that rarely cause infections or using a non-human adenovirus. Examples of viral vector vaccines are AstraZeneca and Johnson & Johnson vaccines. We have discussed the definition of a vaccine, description of an ideal vaccine, and the types of vaccines. Thank you very much for your patience throughout the entire presentation.